Welcome back to another video on design of experiments and analysis of variance. The main problems we need to address through proper design of experiments are bias and variance. Bias is when the results of an experiment are systematically pushed in one direction, usually because something in the way we designed or conducted the experiment is influencing the outcome. To eliminate bias, we can include a control group for comparison. Use randomization to assign treatments, so we're not picking who gets what based on assumptions, and sometimes apply blinding, where participants and even experimenters don't know which treatment is being used, so their expectations don't affect the results. Variance, or sampling error, is the natural variability in data that can make results unclear or less reliable. To reduce this variability, we use replication, running the experiment multiple times so we get a more accurate average. Balanced designs, making sure each treatment has the same number of observations so no group is over or underrepresented, and blocking, where we group similar experimental units together, like using the same machine or operator within a group, to reduce noise from known sources of variation. All of these techniques work together to help us design better, fairer, and more informative experiments, which leads to more confident engineering decisions. Randomized complete block design, in short RCBD, is a powerful experimental design used to control variability from nuisance factors. In many experiments, external conditions or variables that are not of direct interest, called nuisance factors, can introduce unwanted noise into the results. If these effects are not addressed, they can mask the true differences between treatments. RCBD helps reduce this noise by grouping experimental units into blocks that are similar with respect to the nuisance factor, allowing for clearer comparison of treatment effects. The RCBD is an extension of the paired t-test idea. The paired t-test is typically used when comparing two treatments under conditions where each subject or pair can act as its own control. RCBD takes this concept further by allowing more than two treatments, while still controlling for variability due to the nuisance factor. Each block represents one set of experimental conditions, and each treatment appears exactly once in each block. For example, if you are testing 3 types of fertilizer treatments on 5 fields blocks, each field would receive all 3 fertilizers in a randomized order. In this setup, the randomization is done within each block, meaning that the treatments are randomly assigned to the units in each block to eliminate systematic bias. This design ensures that treatment comparisons are not confounded by block-level variability. The statistical model for RCBD includes terms for the overall mean, the treatment effect, the block effect, and random error. Because blocks help account for some of the variability in the data, the error variance is reduced, increasing the power and sensitivity of the test. The table illustrates the data structure of an RCBD. Each row corresponds to a treatment, and each column to a block, and each cell represents an individual observation. Totals and averages are calculated across treatments and blocks. The hypotheses tested in RCBD are the same as in traditional ANOVA. The null hypothesis states that all treatment means are equal, while the alternative suggests that at least one treatment is different. Here we can see how the ANOVA is extended to the randomized complete block design by partitioning the total variation in the data into 3 distinct components: treatment variation, block variation, and residual error. This breakdown allows us to better understand not only whether treatments have an effect, but also how much of the variation in our data is due to blocking, accounting for nuisance factors, and how much is random error. The total sum of squares, SST, measures the overall variability in the data and is split into 3 components. 1. Sum of squares for treatment, SS treatment, representing variability due to differences between treatment means. 2. Sum of squares for block, SS block, representing variability due to differences among the blocks. 3. Sum of squares for error, SSE, representing residual variation not explained by treatments or blocks. Each sum of squares is then divided by its respective degrees of freedom to obtain mean squares, which are the variance estimates used in the F-tests. The mean square for treatments is obtained by dividing the treatment sum of squares by the number of treatment levels minus 1. Similarly, the mean square for blocks is calculated using the number of blocks minus 1, and the mean square for error uses the product of the degrees of freedom for treatments and blocks -1 each. Expected values of the mean squares are shown at the bottom right. These expectations help us understand the statistical properties of the estimates under the null hypothesis. Specifically, the expected mean square for treatment includes the variance of the random error and the treatment effect, while the expected mean square for blocks includes the block effects. The error term is assumed to reflect only random noise, and thus its expectation equals the random error variance. Finally, the formula at the bottom shows how the total degrees of freedom, A, B, 1, is partitioned into treatment, block, and error components. This degree of freedom partitioning ensures that all variability in the data is accounted for and that the F-test for treatment effects is valid. In short, this framework helps us isolate and test for significant treatment effects while controlling for variability due to nuisance factors. This table is a blueprint for performing ANOVA in a randomized complete block design. It breaks down the total variation observed in the data into 3 components: treatment, blocks, and error, and provides formulas for the sum of squares, degrees of freedom, mean squares, and the F-ratio used to test statistical significance. It shows how to calculate the F-statistic for testing treatment effects, while adjusting for block effects to reduce variability. The F-test statistic is used to determine if treatment means differ significantly. It compares treatment variation to error variation and follows an F distribution with A-1 and A-1 times B-1 degrees of freedom, where A denotes the number of treatment levels and B denotes the number of blocks. If the F-ratio is significantly large, it suggests that at least one treatment is different from the others. This structure ensures a more reliable and sensitive analysis, especially when nuisance factors are present. Let's go over an example together. An experiment was performed to determine the effect of 4 different chemicals on the strength of a fabric. These chemicals are used as part of the permanent press finishing process. 5 fabric samples were selected, and a RCBD was run by testing each chemical type once in random order on each fabric sample. In order to conduct ANOVA manually, we need to find the total sum of squares, the treatment sum of squares, the block sum of squared, and finally the error sum of squares. Knowing that we have 4 treatment levels and 5 blocks, we can find the mean squares for treatment, block, and error. Last, we can find the F-test statistic by reporting the ratio of mean square of treatment and mean square of error. Because F-test statistic 75.13 is greater than the critical F value of 0.01, with 3 and 12 degrees of freedom 5.95, we conclude that there is a significant difference in the chemical types so far as their effect on strength is concerned. There is a need for pairwise comparisons and adequacy check for our analysis. Alternatively, we can use Python to perform the ANOVA analysis. By default, the ANOVA output includes an F statistic for the blocking factor. However, this is not the primary focus of our study. Our main interest lies in assessing the impact of treatment levels, in this case, chemical types, on the response variable. Based on the ANOVA results, we conclude that there is a statistically significant difference among the chemical types in terms of their effect on tensile strength. This finding warrants further investigation through pairwise comparisons. To identify which specific chemical types differ, we conducted Tukey's honestly significant difference test. The results show that chemical type 4 is significantly different from the others, indicating it has a distinct effect on strength. Finally, we validated the assumptions of the ANOVA model through an adequacy check. The normal QQ plot confirmed that the residuals are approximately normally distributed, and the residuals versus fitted plot showed no clear patterns, supporting the assumption of constant variance. These checks reinforce the reliability of our conclusions. Thank you for watching another video on design of experiments and analysis of variance. See you in the next one.